Now let's look at optical sensors, and in particular here, a particular example here is your pulse oximeter. So an optical sensor for the pulse oximeter, the optical sensor here consists of two photodiodes. So one that emits light in the red part of the EM spectrum and the other is in the IR part. So the light that passes through the tissues to a photodiode detector placed on the other side is converted into a voltage, which is then filtered, amplified, and digitized. So the pulse oximeter actually operates on a similar principle as spectrophotometers. So you have here a light source passes through your sample, and then you have your receiver, the one that receives the light source. So, how does it actually works, and how does light allows us to measure the blood oxygen levels? So, just a brief recap. So, the oxygen in the blood is actually dynamic. So, this is the movement of oxygen from the tissues in the lungs when you inhale. So, this is in the pulmonary circulation. So, the oxygen in the lung tissue, it enters, this is your alveola, and then connected just through just a single cell layer, the alveola, and then another cell, of course, basically two layers, but two cell layers. But rather you have your lung tissues is very very thin, and then you have the capillaries in each alveola. So it diffuses, the oxygen diffuses from the inside of the alveoli to your oxygen, rather to your blood cells. So here is your capillaries. Now the proteins in the red blood cells, in particular this is hemoglobin, this is the one that absorbs oxygen. And the absorption of oxygen, or rather when hemoglobin picks up oxygen, it actually changes the color. So that's why a vibrant red color of blood, it indicates a very oxygenated, or a blood that is high in oxygen content. So the normal blood oxidation levels, we quantify them in terms of the saturation of the peripheral oxygen, or SpO2. So when it enters, or when the hemoglobin picks up the oxygen, the hemoglobin in the red blood cells pick up the oxygen from the lung tissues. So you have a high SpO2 value. So as the blood circulates throughout the body, the oxygen is being distributed to the peripheral tissue. So here is schematic B. So this is your capillaries, and this is the peripheral tissues, the tissues in the rest of your body. So the hemoglobin, which is the carrier, it releases the oxygen, and oxygen diffuses to your peripheral tissue. So the SpO2 here is a bit lower. So the blood that directly comes from the heart, after the arterial blood flow, you have a high SpO2 here, and the slower venous blood flow, it has a lower SpO2 level. So the SpO2 represents the percentage of hemoglobin in the arteries that is oxygenated. That means it's the number of hemoglobins that is carrying oxygen. So here is the calculation of the SpO2 level. So the percent, as a percentage, this is the concentration, or the amount of hemoglobin that carries the oxygen, that's HbO2. And this is the total number of hemoglobin, whether it carries an oxygen or not, in the form of Hb. So this is the total hemoglobin. So the number of hemoglobin carrying oxygen over the total amount of hemoglobin present. So this is your SpO2, just a simple percentage. Now, the pulse oximeter, I think you have seen as an example, you might own one, especially now in the times of pandemic. This is a very important device, especially for those patients with COVID, because their oxygen in the blood can fall drastically, and actually one of the main causes of death. Well, anyway, the pulse oximeter, it releases two types of light, the red light and the IR. Now, the reason why it must have two types of light is because the absorption of light by oxyhemoglobin, that is the hemoglobin that carries oxygen, and the oxyhemoglobin, this is the hemoglobin that does not contain the oxygen, are different, or rather they absorb light at different spectrum. So here is the EM spectrum. and your absorption. So, ang oxyhemoglobin has a slightly lower absorption. It absorbs red light and it peaks at around 900 to 950 nanometers. So, this is around the red region. And then you have here the oxyhemoglobin peaks at, actually it's not exactly a peak, but the range possible is at the IR range. That's why you have two lights. One, for, the red light for picking up the amount of oxyhemoglobin, and the other for picking up the oxyhemoglobin. So that the SpO2 can therefore be calculated from this general equation. So this is the reason why you have two lights there. And if you look at the this SpO2, you have actually a red light inside. If you pick in at your uh, light. So you have here your the red and IR light is again transmitted through the finger and the photodetector. So as the light passes through the finger, so if you have the oxyhemoglobin in the finger, absorbs some of the red light. So the amount of the red light produced is reduced, and you have your reduced when it goes to your photo detector. And then you have the again the infrared, the same parameter. So you have your photo detector again also getting the signal from the IR spectrum. This is now for the oxyhemoglobin. So this is also the absorption for the water and lipid. So the problem here is at lipid. Lipid is absorbed again at it actually has a peak absorption at the red spectrum, similar to the peak absorption of your red or the oxyhemoglobin area. So the lipids, the amount of lipids can pose a challenge or a problem. So you have that. Okay, so and then for correcting this error, we just put it in the or consider it in your calculations later. So how do you measure your SpO2? Or how does actually optical sensor senses the oxyhemoglobin levels? So HbO2, this is oxygenated hemoglobin, and the hemoglobin concentrations. So they are measured by applying the Beer-Lambert's law. So the Beer-Lambert's law directly correlates the intensity of light with the concentration. So here is the measured light intensity. This is the incident light, the light from the source, and then this is the light intensity. So you have the absorption coefficient, and which is a constant, and your path length, the amount of the path it follows to the tissue. And lastly, you have C as the concentration. So the intensity of the incident light is assumed to be proportional to the current through the LED device, and the current generated by the photodiode to be proportional to the intensity of the transmitted light. So you now have the idea on how to get your concentration through actually this is an application of the application here is very similar to the application of UVV's spectrophotometers. So the incident light, the measured intensity of transmitted red light at 660 nanometers is given by the differences in the tissues, hemoglobin, and oxygenated hemoglobin. But there's a caveat or there's a problem here because in a heartbeat to heartbeat basis, even in the pulse in your fingers, so there's a difference between the amount of SpO2 in the arterial blood, which is oxygen rich, and those at the venous blood, where the blood has already, the oxygen has been distributed to the rest of the body. So there's a signal difference at each heartbeat, at each pulse rate. So these differences should rather is accounted for by taking the ratio of the maximum and minimum intensities, because again at each pulse you will have a peak, a very high SpO2 level, and then it goes down immediately at each heartbeat. So to get to correct this problem, if you want to get the average or the actual concentration, just get the ratio of the intensities of the heartbeats, or rather sorry, the intensities of the light. So the red and IR light intensities are calculated using these values. So this is for the red light and this is for the infrared light. And then getting the IR pulse modulation ratio, this is just the ratio of the ln of the red light to the IR. You can now apply it into this equation, which is actually quite complicated equation. What is usually done in reality, in actual practice. So this is for commercial use, which is not necessarily must be accurate, but must be good enough to have a good determination of the blood oxygen levels. Because this one is highly accurate but very complicated to use, you will have a higher computational demand here. But for normal commercial use, you have it's simply they you simply use a quadratic relation to get your SpO2, where the coefficients A, B, and C, they are constant, obtained by fitting your second order polynomial to a graph of values. So if you notice, when you first apply the SpO2, there's a lag time between attaching the SpO2 versus um, attaching it in the or rather getting the readout for your blood oxygen levels. That is because the oximeter is actually calculating your SpO2 level from the light intensities. So if it's a commercial one, the lag time is due to the to first getting a graph, a data, sets of data, and then fitting a graph there, and then getting the A, B, and C from that. And that is also why calibration of individual pulse oximeter is necessary. So when you want to have a really accurate pulse oximeter, make sure that when you buy one, they have a certificate of calibration. So this is the signal usually seen in your um, the red light. So you have this is for the IR light and this is for the red light. So the timing diagram, so these are the two LED lights are alternately switched on and off for 25 microseconds with a 250 microsecond off time to measure the string light component subtracted. So basically signal processing of your photodiode detector. So this is a schematic of the semiconductor photodiode detector. So circuit representation, so you have IL is the current produced by the incident light, ID is the diode current, CJ is the junction capacitance, you have RSH is the shunt resistance, RS is the series resistance, IO is the shunt resistance current, and I out the output current that passes to the filters and amplifiers in the next stage of the device. So here is your plot of sensitivity of your instrument. So this is the output current, responsivity in amps per input power, and the function of the wavelength for a typical photodiode. And then the raw signal is a jagged line because this varies from heartbeat to heartbeat. So that's it for optical sensors. So in the next part, we will talk about chemical sensors.